Welcome to The Explainer. Today, we're diving into a chapter that's not just a big deal on your exam, but is literally changing the world as we speak. Biotechnology. We're going to break down the key principles and processes, step by step, to give you everything you need to absolutely ace this section in your Class 12 boards.
So, let's start with the big question. What even is biotechnology? Well, you can think of it as having two different personalities. On one side, you've got the traditional stuff we've been doing for centuries, using microbes to make curd, bread, or wine. That's the old school view. But the modern view, which is what we're really focusing on for your exam, is on a whole other level. Here, we're talking about genetically modifying organisms to get them to produce exactly what we want on a massive scale. We're talking about high-tech things like DNA vaccines and even test tube babies.
All right, to really nail this topic, here's our game plan. We'll start with the core principles, the absolute foundation. Then we'll open up the genetic engineering toolkit and check out all the cool molecular tools scientists get to use. After that, we'll walk through the actual step-by-step process of creating recombinant DNA. And finally, we'll see how you take something made in a lab and scale it up for the real world.
Okay, let's get right into it with the core principles. Everything we do in modern biotech, every single breakthrough really comes down to two fundamental techniques. If you can get your head around these two pillars, you've basically got the key to understanding the whole chapter.
Think of it like this. Genetic engineering is the designer. It's where you go into the actual blueprint of life, the DNA or RNA, and tweak it to change what the organism looks like or does. But a great design is useless if you can only make one, right? That's where bioprocess engineering comes in. This is the manufacturing arm. It's all about creating the perfect sterile environment to grow massive quantities of your newly designed cells so you can produce things like antibiotics or vaccines on an industrial scale. One designs the change, the other makes it happen.
So where did this all begin? It's a pretty cool story, actually. Back in the early 70s, you had these two scientists, Herbert Boyer and Stanley Cohen. Boyer was studying these amazing enzymes in E. coli that could cut DNA at specific spots, leaving these things he called sticky ends. At the same time, Cohen was getting really good at taking tiny rings of DNA called plasmids out of bacteria and putting them back in. The light bulb moment came in 1972. They decided to combine their work. They used Boyer's enzymes to cut out a gene for antibiotic resistance and pasted it into one of Cohen's plasmids. And it worked. They created the world's very first piece of recombinant DNA. A simple cut and paste that started a revolution.
Okay, so Boyer and Cohen showed us it was possible. But to do it yourself, you need the right set of tools. And for your exam, knowing this toolkit inside and out is absolutely crucial. Think of it like a super high-tech workshop. So let's open up the toolbox and see what's inside. First up, the real stars of the show, restriction enzymes. Yeah, you can call them molecular scissors, and that's a great way to think about them. They scan along a DNA molecule looking for a very specific sequence of letters. And when they find it, they make a cut. Simple as that. And a great fact to remember for your exam, the first one ever properly identified and understood was called Hind-2. Let's get a closer look at how these scissors actually work, using a famous one called EcoRI. Now, EcoRI is always on the lookout for a specific sequence, GAATTC. And here's the cool part. The sequence on the opposite strand, read backwards, is exactly the same. That's called a palindromic sequence. Now look how it cuts, not straight through, but in a staggered way between the G and the A. This leaves these little overhangs of DNA, and we call them sticky ends. Why? Because they're ready to stick to any other piece of DNA that has a matching sticky end. It's perfect for pasting new DNA fragments together. Now, here's a quick distinction that often comes up in exams. Restriction enzymes are part of a bigger family of enzymes called nucleases. There are two main types. You have exonucleases, which chop off nucleotides from the ends of the DNA. Then you have endonucleases, which make cuts at specific spots within the DNA. Our restriction enzymes are a type of endonuclease. Just remember, exo for trimming the outside edges, endo for cutting somewhere in the middle. Okay, you've used your molecular scissors to snip out your gene of interest. Now what? You can't just throw it at a cell and hope it gets inside. You need a delivery vehicle. We call this a cloning vector. The classic analogy is a mosquito carrying the malaria parasite. The mosquito itself doesn't cause the disease. It's just the vector that delivers the parasite into a person. In the same way, we can use a plasmid as a vector to carry our alien piece of DNA into a host organism. And here is a legendary workhorse of genetic engineering, the cloning vector PBR322. This diagram is pure gold for your exams. Let's break it down. See where it says ORI? That's the origin of replication. It's the start copying signal for the cell.
Then you see AMP-R and TET-R. These are selectable markers. In this case, they give resistance to two different antibiotics. We'll see why that's so useful in a bit. And ROP codes for proteins that help control how many plasmid copies are made. It's an incredibly clever bit of engineering.
So, to sum it up for an exam answer, a good cloning vector needs three essential features. First, it needs an origin of replication, or ORI, so it can be copied by the host cell. Second, it needs a selectable marker, which helps us find the cells that actually have our vector. And third, it needs cloning sites, those specific spots where we can insert our foreign DNA. Get those three things down and you're all set.
All right, we've explored the toolkit. Now it's time to put on our engineer hats and actually build something. We're going to walk through the entire process of creating recombinant DNA from start to finish.
The whole process, known as recombinant DNA technology, or RDT, can be boiled down to these 5 main stages. First, you have to isolate the DNA. Then you cut it up. Then you make millions of copies of the specific gene you want. Next, you insert that gene into your host cell. And finally, you get that host cell to produce whatever it is you've programmed it to make.
So step 1 is getting the pure DNA. To do that, you have to break open the cell walls, maybe with lysozyme for bacteria or cellulase for plants. Then you use other enzymes to get rid of any contaminating proteins or RNA. The last part is super cool. You add chilled ethanol to the mix, and because DNA can't stay dissolved in it, it suddenly appears out of the solution as these fine white threads. You can even collect them by winding them onto a glass rod in a process called spooling. Usually, you only start with a tiny amount of your desired gene, which isn't nearly enough to work with, so you need to amplify it. And for that, we use a technique called PCR, or polymerase chain reaction. It's basically a molecular photocopy machine. It works in a 3-step cycle that repeats over and over. First, you heat the DNA to separate the 2 strands. That's denaturation. Then you cool it so small DNA primers can bind. That's annealing. Finally, a special heat-stable enzyme adds new nucleotides to build a new strand. That's extension. Repeat that cycle 30 times, and your one copy of a gene becomes over 1 billion copies.
Now comes a really tricky part. How do you get your new recombinant DNA into the host cell? The cell membrane is pretty selective about what it lets in, so scientists have come up with some clever workarounds. For bacteria, you can use something called heat shock to briefly make them porous. For an animal cell, you can use a tiny needle for microinjection, literally injecting the DNA right in. For tough plant cells, there's even a gene gun that fires tiny gold particles coated in DNA straight through the cell wall. And sometimes you can even use a disarmed virus as a delivery vehicle.
So, we've done it. We've successfully made our genetically modified organism in a lab. That's a huge step, but it's still just in a tiny test tube. To actually make enough product to help people, we need to think bigger, a lot bigger. How do you go from the lab bench to an industrial scale? The answer is a bioreactor. We're talking about huge, 1,000-liter vessels. A bioreactor is basically a luxury resort for your cells. It provides the absolute optimal conditions for them to grow and produce your protein. The perfect temperature, pH, nutrients, and oxygen levels. On the left, you can see a simple stirred tank reactor. The one on the right is a sparged stirred tank reactor, which bubbles sterile air through the culture to make sure all the cells get plenty of oxygen. Okay, the bioreactor has done its job, but we're still not finished. What you have now isn't a pure product. It's a huge tank of cellular soup with your product mixed in with all sorts of other stuff. This final, critical stage is called downstream processing. It's the whole cleanup operation. You have to separate your product, purify it to an incredibly high standard, and then formulate it with preservatives so it's ready for the market. This is a huge and often very expensive part of the whole process. So, as you study for your exams, try to see the big picture. Don't just memorize the individual steps. Understand how it all flows together as one continuous process, from a single gene all the way to a life-changing product. Thinking about the whole journey is what will really make your answers shine. And beyond the exam, it gives you a real appreciation for the sheer power and complexity of this amazing field.